Our speaker for today is Dr. Chandra Sekhar Pedamallu. Dr. Chandra Sekhar Pedamallu comes with extensive experience in bioinformatics and computational biology, specializing in cancer genomics. Before joining Excelra, Chandra was associated with Sanofi Incorporated, identifying novel drug targets in oncology, molecular, and immuno-oncology. Prior to Sanofi, he was at Dr. Meyerson Lab, Dana-Farber Cancer Institute, and a visiting scientist at the Broad Institute of MIT and Harvard. During his 6 year tenure at Harvard, he led microbial analysis, pathogen discovery projects in cancer, and other diseases. He has contributed substantially to over a dozen projects under the auspices of the Cancer Genome Atlas, or TCGA. Chandra holds a PhD in systems engineering, his area of research being global optimization, and he got his PhD from the School of Mechanical and Aerospace Engineering at NTU Singapore. He has co-authored over 85 manuscripts published in highly reputed journals, including Nature Genetics, Science, Cell, PNAS, etc., and has been inducted as a Fellow of the Royal Society of Biology. Thank you, Chandra, for joining us. The stage is all yours.
Today's topic is on artificial intelligence and machine learning approaches for tumor detection. So as Kunal mentioned, I'm a Vice President of Bioinformatics and Scientific Head at Excelra. So before going to the topic, I just want to give a brief introduction about Excelra, where I'm coming from. So let me see if I can. Yeah. So Excelra is a leading solution partner to accelerate life sciences innovation by providing and offering solutions both in the data as well as analytics. And it is a seamless unification of domain and also informatics. Excelra comes with 2 decades of experience in the life sciences market. We work with 16 out of the 20 pharma companies, having 175 plus active customers. And also we have a strong agile team of over 1,000 people now. They are specialized both in life sciences and technology. And of those 1,000 people, we have 120 people that are bioinformaticians. And that makes Excelra as a bioinformatics powerhouse. And we are having a headquartered in India and having offices across the world, that's in US, UK, Belgium, Netherlands, and Germany and Switzerland, where we also have consultants that work from there. So if you look at Excelra at a very high level, there are 3 main pillars that we have it. The first one is on data. The second one is on R&D, IT, and data analytics. Under the data wrangling that we've shown here, we provide services for structuring the data, harmonizing the data, and also managing the data and subscribing the data. And if you come to the data analytics, that is our domain, where we have bioinformatics, HEOR, value evidence, as well as statistics. Whereas coming to the R&D IT, which is the informatics point of view, we provide services in cloud, DevOps, as well as data engineering and visualization. And we also do ELN and LIMS configuration as the customizations. So if you double click on these 3 pillars, what kind of services we offer under the data wrangling is, in a very high level, it is the data curation and harmonization by using the machine-assisted technologies that starts from preclinical to clinical. And also we develop automated ETL pipelines to do the data curation and harmonization. Same time, we also follow the data verification principles, and we based on that. And also we have a semantics technology to build knowledge graphs from the data that's been generated. And the second pillar that I talked about is an R&D IT, which is a bio-led IT solutions that's primarily to integrate and manage the data by providing cloud infrastructure, migration and modernization. As I said, we do a lot of data management, both engineering aspects of it as well as the visualization and AML. And we also have consultants that does ELN LIMS consulting, as well as implementation integration, and also software development. Under the domain aspects of it, which comes under the data analytics, we deliver insights through computational methods. We work with multiple range of omics data. That's all kinds of omics data that you can think of by doing the data management as well as analysis. And also we build applications that helps from the discovery to the clinic. So across the drug discovery value chain, we have a footprint as a data analysis, data experts from there. And we are consultants providing consulting services as a scientific consulting people. So in addition to the services that we have we also have products that are listed here. The first one is a GoStar, which is a small molecule activity relationship database, which contains about 9 million molecules. And we have a GoBiome, which is a biomarker intelligence database. And also we have an OP Square platform, which is an NGS data analysis platform, where you can upload your FASTQ files, analyze the data. At the end, you're going to get your output. And in addition to that one, we have an Immune Repertoire platform, which is primarily geared towards the immunogenomics type of analysis, which is kind of a TCR, a BCR analysis that you can do it. In addition to that one, we have an AWS management application. The primary role of this particular app is to manage, optimize the AWS usage. And we have a single cell browser, which helps to visualize the single cell data that has been generated from your experiments. This is in a nutshell what Excelra is and what kind of services and what kind of products that Excelra comes to the table. We are a great partner with the biotechs as well as pharma companies. Now let me go into the presentation now. The agenda of the presentation is as follows. I start with biomedical imaging, giving you some introduction about it, and I will dive into AI/ML approaches that is used for tumor detection. And I will also show some applications of AI/ML approaches in the tumor detection. And I will end the presentation with a summary. So coming to the biomedical imaging, most of you know that this is a powerful tool for visualizing the internal organs and its diseases. So it has been used widely between the bench to the bed or bed to the bench. So the place where you use this imaging technology can classify these ones into three main categories. One is cellular imaging, preclinical imaging, clinical imaging. Cellular imaging is primarily used at the early stages of the drug discovery pipeline, where it can be used in the target validation, I would call it as, where you try to visualize a cell population. It can be a single cell, it can be subcellular structures. These are some of the examples that are listed here are being used as a technologies for that. In case of preclinical imaging, primarily used for living animals for research purposes, that you can see it here where we can use it in different aspects of it in the animal models. So some of the examples are listed here, which is the micro mRNA, micro CT, and a micro PET. And in case of the clinical imaging, that is primarily what we're going to talk today, is performed to both for the initial diagnosis as well as to identify the disease and their follow-ups by the radiology team. So some of the examples that you can see is an MRI or a PET scan or an Excel. So let us, so the whole presentation that we're going to talk here is only in the clinical imaging point of view. So let me double click on the clinical imaging and see what are the different imaging modalities that one can have it. Here, the imaging modalities are divided based on the kind of the data that is going to be capturing. It can be an anatomical data or it can be functional data. So for example, if you take X-ray, it is a pure anatomical imaging modality, whereas a PET scan that you're seeing here is a more functional data. It can be like a biochemical or a metabolic data that is going to be captured. Whereas the rest of the modalities that you're seeing here, which can be an MRI or optical, they are capable of capturing both anatomy as well as functional, based on the protocol that we normally use it there. So each of these imaging technologies that we are talking here comes with pros as well as cons. One of the widely known con for some of these technologies is the radiation risk. And that can be mitigated in other cases. Like for example, I would say in case of, I think in optical ones, the amount of risk for radiation risk is much lower. So as we know that imaging plays a vital role in many modalities. At also same time, it plays a vital role in a lot of phases of the cancer management. Let us see what are the different phases of cancer management that you can think of. In a very high level, it's a 2,000 feet level, what you can bin the cancer management phases into 5 here, starting with screening, ending with understanding and recurrence of a cancer. So these are the different cancer imaging modalities that have been used wide across. So you can say, like in a screening case, you use mammograms, colonography, whereas in case of diagnosis to the cancer recurrence, you use normally a PET scan, it can be a CT scan or an MRI. And some cases you use X-rays. This is not extensive list. I just give you some examples that you can see it here. So then let's step back and see, this is what is being used as a modalities in the cancer management phases. What happens in the clinic when a patient visits to the clinic? So this is a typical patient journey that you can think of from visit to the diagnosis. So once a patient comes to the clinic, a clinician or an oncologist perform a clinical examination. And in cases that are relevant, they take a biopsy of this tissue, and then they refer the patient for a lab and imaging test. Once they refer them to the lab and imaging test, the patient goes to the lab to get lab tests that includes a diagnostics test. And also same time, they also go into the imaging. So where a radiologist are going to perform a radiological imaging, and based on the images that are coming out of it, they're going to create a report. Both the lab test and the radiology reports are being given to the oncologist to help them to make the next steps that will be planned for this particular patient, which direction they want to go and what kind of treatment they want to give to these patients. So now we're going to focus only on the step 4, where a radiologist performing the radiological imaging and also making these reports. What are the kind of the challenges that these radiologists face? Here are some of the challenges or in the context of radiology. So often the field faces is a scarcity in experienced radiologists, and also the detection accuracy always depends upon the radiologist experience. At the same time, it takes a longer time periods because especially in the Western world, the number of radiologists that are available to hospitals are fewer. Sometimes the patients has to wait until they get those reports. At the same time, sometimes the data can be misinterpreted if the radiologist is not experienced enough for that. And also it takes a lot of time because it has to go through a lot of images at the same time. They have to write a report. It's a laborious process. So what does AML can bring to here is that it can bring approaches that can assist these radiologists in both the tumor detection as well as diagnosis. So let us see what is an AML, right? Very, very high level. Artificial intelligence is something that can stimulate the human thinking and behavior, whereas a machine learning is a subset of AI that can provide you the classification or prediction with or without humans supervision by learning from the data that has been there. So one advantage that the field of medicine is having is because of the digitalization, because most of the CT scans, X-rays are in the form of images. So they became early adapters of this AML. So by using the AML approaches, one can do abnormality detection. At the same time, they can do a characterization of suspected lesions. So in a nutshell, what I can say is that the recent advancements that we have it in both biomedical imaging as well as in AML approaches provide an unprecedented opportunity to do the tumor detection and diagnosis. So let me go into the tumor detection. What is a typical pipeline looks like when anybody wants to identify whether this particular tissue is a benign tissue or a tumor tissue or a malignant tissue. So here is the high-level workflow for tumor detection. There are 3 main steps in this workflow. The first step is to build the model, and the second step is to validate the model, and third step is use in the real time. So the first and the foremost thing that people will do is that they try to get the images from the public domain, and then they categorize or classify those data into 2 pieces. One is the training data, another one is the validation data. Once you have a training data with you, you're going to run through certain core tasks that are listed here. The first one is the image pre-processing, and the second step is feature selection, and the third one is model fine-tuning. In an image pre-processing step, what people normally do is that they read the images, they resize the images, they remove the distortions of the images, and sometimes they do segmentation also on the images. And once they make these images ready for feature selection, once they extract out the relevant features from there, They build the model and they try to fine-tune the model so that the performance of the model is good enough. That will help to build a classification model for you, because this has been trained on a training data that can lead to a training prediction model with you. So now you have a model with you. Now you want to know what is the performance of this model. At that point, you're going to use your second data that you have it, that's the validation data. So the validation data, you take it, you send this validation data through image pre-processing. And once it processes the pre-process through the image pre-processing step, you're going to run through this through the trained prediction model. From there, you will know what tissue is a benign tissue, what tissue is a malignant tissue. That will also help you to understand what is the performance of your trained prediction model now, because we already know the labels of them. So by looking at the performance of the metrics, you're going to say, yes, this model is pretty good. Let me use it in the real time. If not, then you need to go back and you try to fine-tune the model so that you'll get a better model at the end of the day. So you take that model, you implement in the tumor detection, where a patient in real time comes, you take the image, you send that image through pre-processing module, and then you ran that pre-processed images through the trained prediction model. Then it's going to give you whether the tissue is a benign tissue or a malignant tissue. If that is a malignant tissue, then you're going to go to the next steps of the clinical, the next steps in the clinic. So now we know the high-level workflow of a tumor detection. Then if we can drill down and say, how does it work really? So the first thing that happens is that you take the image, you undergo several rounds of convolution. So that means that you're going to give a convolution and pooling operations so that you can go to extract out the high order features from there. So here you can see stride and filter sizers, and then that can makes your maximum pooling across the tails that creates the feature maps. And then you take the feature maps and then going to reshape them into a 1D vector. And then that 1D vector will be fed into your connected layers. And the connector layers will help you to do the final prediction, whether it's a tumor or a normal. So if you double click on this particular box, that is your neural network box that you have it. It contains two primary things. One is a kernel function, another one is an activation function. At each layer that you have it here, there is a weights that are applied to the kernel. And that will resulting values will be run through your activation functions. Once you went through this activating function, the final layer is a softmax function here that has been applied so that it will generate a probability of your output class. So if you run the same algorithm, same steps on known data, there's a training data, you know what is the ground truth labels so that you know what is the error of your predictions. So this is how a typical CNN layer model works in the case of images, that biomedical images that you get it. So let us now think about what are the things to be considered before you build these models. The first thing is that the training data, because one thing we need to keep in mind is that the prediction efficiency of these models, the trained models, is always dependent on the training data. So you need to have the volume and diversity of the data. At the same time, you want to have a very good quality data, good resolution data, as well as different modalities. So one sample training data set that I'm listing here that is coming from Lee et al. Paper that's been published in 2023 that contains multi-organ data that is already there. And also beyond from this sample training data, there's a lot of data that is out there, which you can use it to train your model so that you can get a better prediction efficiency from that. Okay. So let me move on from there and say that what are the frequently used models in biomedical imaging? So for example, in case of the classification imaging task, that means that classifying a disease or a condition from medical images, normally we use CNN models. And also the last function that has been used is a cross control. And also for the evaluation metrics, people use accuracy, F1 score. And there are other steps that are also involved in this processing of this data that includes a segmentation, detection, and registration based on need basis. So for example, segmentation is primarily used for segmenting organs or tumors or structures in a medical images. In case of detection, they are primarily used detecting objects or abnormalities in medical images. Whereas in case of registration, the primary task that people use it for registration is aligning and spatial transformation of medical images, because you take multiple images during the process of going through these taking the images at the radiologist place. So from there, they want to identify which ones are the better ones to do your prediction models. And there are a lot of loss functions that people use it for different steps, and also the frequently used models here and their evaluation metrics. So let me step forward and say, okay, fine, now let me find out what are the applications of these AML approaches in the context of tumor detection. Are there any applications that are out there? So I'm giving you two examples here. One is on melanoma is one example, and the other one is a lung cancer. So in case of a melanoma, here the primary objective here is to classify skin cancers, images that have been provided, and the data modality that's been used is a dermoscopic images. Dermoscopic images are kind of the imaging techniques that are widely used primarily to enable the skin surface that is visualized by the light amplification immersion fluid. So the training data that's been used in this case is a ISIC 2019 data set, and the ML model that has been used here is a deep neural network model. So if you look at the workflow that is listed here, right? So the data sets are taken, you went through a pre-processing step. Once you have a pre-processing step that is divided into training data and test data, and then you go through a segmentation using UNet, and then feature extraction is done on those segmented data using SpeezeNet, and then you do a parameter tuning. That's the model tuning that you do it by using whale optimization. Then you have a classification model with you. Then they ran the performance evaluation by using test data. But the same neural network that you have it here, or same model, can be run on the real-time data also. At least by looking at the test data that you have it in hand, they achieved about 99.9% accuracy, and also have a average F score of 0.99. It's pretty good. So this is one example that I can say that is coming from the deep neural network that is used for doing the skin cancer classification, which is melanoma classification. So if we go to the next application, that is a neural network that has been built for lung cancer classification. Here, the objective is the same like skin cancer. We want to understand how to classify these lung cancer images, and the modality that we have used is the CT scan images. If you see the modality from previous to this one, is the data modalities are different here. So the training data that has been used is the cancer imaging archive, and they used a deep learning with an instantly trained neural network for this. So if you look at the workflow, it is very similar to skin cancer. You take the images, you ran through the pre-processing step, segmentation step. Once you went through the segmentation step, you're going to extract the features from there, and then you take the features to build your neural network. Once you build your neural network, that neural network model is going to be a trained neural network, because you already went through a large cancer imaging archive. Because you have divided them into training and test data set, you can test the performance of this. So the performance metrics looks phenomenally good, which is 98.4, and also average F score is about 98.98. So we have a pretty good models, right? So why people are not using it in the clinic? So there is always one thing that we have, it is acceptance of these of these AML approaches is a bigger challenge for people, or for people to use them in the clinic. So let me step and also go forward and say that, are there any value addition studies that have been the value addition, meaning that AML value addition studies that have been published in the literature? Here are two examples I'm showing here. One is about a standalone artificial intelligence technique that has been used for breast cancer detection. Here, the modality they use, the data modality, if you see it, is a mammography for this one. It is not like a CT scan. So if you can see that application of AML across different data modalities that you're seeing, the data imaging modalities that you're seeing here. So here the primary goal is to see how well this particular AML approach is going to do when you compare with a radiologist. Are they doing similar? Are they doing are they performing inferior when you compare with a radiologist? So they created a panel of 100 radiologists, and they have this breast cancer mammograms with them, and they went through radiologist one process as well as the AML technique they have developed it. So one thing they have found from this study is that the AML approach that they have developed it was statistically non-inferior to the average of 100 radiologists. That means that the approach can be used in the clinic, or it can be used as an assistance to the radiologist. That can reduce the effort that radiologist is putting on pulling together these reports, analyzing these images that are coming out of these instruments. Because often these instruments can be very good and they produce a lot more images. So I think there is a need for more radiologists, right? So what they have also performed in this particular paper is that, does it really help a radiologist to save time and also have a potential to implement or not? In this case, what they have found is that there is a threefold improvement in target lesion measurement, concordance with the oncology records that have having it. At the same time, they reduce the radiologist examination as well as interpretation time by 37%. So these two studies that I'm presenting here gives you the value that AML approaches can bring to the table when they use it in the clinic, and at the same time it can help and assist the radiologist. I'm not saying let us replace the radiologist. What I'm saying is that it can help a radiologist to make these creating a report or understanding an image much faster. Okay, so there are a lot of press that is talking about this AML approaches in detection, diagnosis. Some of them are listed here. These are all recent publications that I can say. The first one is about the new AI software that achieves about 100% detection rate from melanoma. This is published in newsmedical.net, which is a very recent one, actually. On 11th of October, they published this one. They claim that this particular software can do a 100% detection rate. And also there's another report that's been published in MIT Technology Review, which is also a very recent one published in last year. So talking about AML that has been used that can help doctors to catch breast cancer. So it looks like this way, also from our own slides, that looks like AML approach can bring value to the doctors as well as radiologists. And also there's another one, promising one that has been published, is about this Cybele, which is a AML technique that is used to identify early signals of lung cancer that doctors cannot see. So it is going beyond what doctors can do. So that's for sure is indicative of how powerful these techniques are. And also if you go into the FDA approvals for AML enabled devices that have been approved by FDA, there is a huge number of devices that are being approved by FDA that are being enabled by AML. These devices are the ones it can be a CT scan that is being powered with AI/ML that helps and assists radiologists to make those reports. And also same time, they can also assist them to help to identify the contours of those tumors. So it is so powerful. So we should better try to adapt them into the clinic and move forward from there. So let me come to the summary of this. So I would say the field of medicine have this luxury because of digitization they have done all these years in the form of both the CT scans, X-rays, and so on and so forth modalities. So that's the reason why they are the early adapters of AML. But the main problem that I'm seeing here is that AML is primarily used as a retrospective, so not a prospective. So the primary challenge that the field is facing now is the acceptance from the radiologists and clinicians. But I think the way that we need to go forward is that the AML approaches can help radiologists, assist these radiologists to prepare, to analyze the images that have been produced. Okay. So in the context of oncology, I would say these well-trained models that you're having in hand can help you diagnosis and classify tumors, and also help you to detect early stage tumors. So early stage tumors are very important because that also changes the way you're going to treat a patient. At the same time, the burden on the healthcare system will be much, much smaller if you can detect these tumors at early stage, and also the prognosis of these tumors will be much, much better in those cases. And by gathering the genetic and histopathological data together, it can also assist in the pre and post-operative plan. And at the same time, it can also help us to predict overall survival outcomes. So in summary, I would say, AML is a transformative technology. We should really adapt them into our clinic and help the clinical practice and come with the better models at the end of the day. So, but the other challenges that we can think about is using the right model to the right data. That's always a challenge. At the same time, data asset creation, because the training is an important piece of the puzzle. So the training data is very important for building any of these models. And also you need to have a high throughput data analytical pipelines with this. So in these three aspects of it, Excelra can be a great partner. As I said, that we are a leading solution partner to the biopharma companies as well as the life sciences. So I would say, we are there, but it's all about acceptance of AML approaches in the clinic changes the course of it. So I will stop here and I will go into the acknowledgement section, which is I would like to thank Sushant, Anu, and Puneet in my group, because these are the people that helped me to pull together this slide deck. And also I would like to thank Matthias, Jitesh, and Ujma, and also Praveen and Ranga and Prashant and Bindu. These individuals are the ones helped me to polish these slides. At the same time, also helped me to connect with ACS to have this seminar. So, and also I would like to thank Jan, Martin, and ASP, Andvir, who is our CEO, and Martin is our commercial head of bioinformatics, and the whole bioinformatics business unit and Excelra family, and also the ACS, the American Chemical Society, for giving this opportunity to talk to you all. And first of all, I would like really thank every one of you joining from different parts of the world. And also thank you very much for your kind attention. If you have any questions, or if you want to learn more about Excelra or our offerings, please contact at this particular email address. Thank you very much, guys.